Hello, class. This is a recorded lecture about the sequence of events involved in transmission at a typical chemical synapse in our nervous system in the brain. And so let's get a few landmarks here. Up here at the top, we have our presynaptic neuron. And down here at the bottom, we have our postsynaptic. And this might be another neuron, or it could be, as we've discussed, an effector cell, such as a muscle. For this illustration, we'll make this postsynaptic cell another neuron. So before sitting here in our, right, and to be clear, this is our axon terminal. Sitting here ready to go before an action potential even occurs, we have synaptic vesicles that have already been pre-filled with neurotransmitter. So these green dots represent our neurotransmitters ready to go. Now these vesicles are bound together via a protein called synapsin. And so they're waiting here in the presynaptic membrane, waiting for an action potential. So when an action potential comes down the axon and into our axon terminal, the local membrane does what? You should know that it depolarizes the membrane. There we have a wave of positive sodium ions coming down into the axon terminal, depolarizing our membrane. Now, this depolarization causes activations of a voltage-gated channel. Now, we've talked about sodium and potassium voltage-gated channels in class before, but we have a new one here that I've mentioned, calcium. And let's represent calcium here as red. Calcium ions that are in higher concentration in the extracellular space. So this depolarization activates these calcium channels, and calcium rushes in. Now, inside or outside of the cell, there's about 10,000 times more calcium. So they easily flow in down their concentration gradient. Now, the sudden rise in calcium concentration is a trigger. First, this calcium mobilizes individual vesicles away from this complex, this vesicle synapsin pool. And it does this by activating something that we can generally call calcium activated protein kinase. And for those of you who have taken cell, this might be familiar to you. This protein kinases is a very general term for a protein that phosphorylates another molecule. So calcium, and let me make things a little bit more clear here. Calcium, what it does is it activates the protein kinase that can then go in, phosphorylate synapsin, which causes it to break up this complex up here, freeing the vesicles. All right, so it breaks up that one, and it would break up this one as well. Disconnecting synapsin from the vesicles, and the vesicle is now free to move down. Down into the near the membrane in our postsynaptic or our presynaptic terminal here. Now, calcium also is not only facilitating the movement of our vesicle closer to the membrane, but it's also activating another class of proteins, of which there are a few kinds, on the vesicle and on the membrane. And these are called snare proteins. And we'll get into these in more detail later. And so calcium is not only breaking up via protein kinase, the synapsin vesicle complex, but it also interacts with these snare proteins here. And what it does is ultimately causes this vesicle to merge. Merge with the presynaptic membrane, and thus spilling out the neurotransmitter into our synaptic cleft. And this has a very special name, this process. This is what's known as exocytosis. So let me write that out here. Exocytosis. This fusion of the vesicles with the presynaptic membrane, releasing neurotransmitter into the synaptic cleft. That whole process is exocytosis. And so neurotransmitters are diffusing across the cleft, and this can take up to a few milliseconds. And what do they do in the postsynaptic cell? Well, there are membranes, membrane-bound receptors that specialize in binding to these neurotransmitters. And as we'll learn, there are different types of neurotransmitter receptors, but in this example, to sort of keep it simple, these are going to be neurotransmitter activated ion channels. A more general term for a receptor that binds to a molecule to activate it are called ligand activated receptors. And so I'm saying that these are, I'll go ahead and write that out here. Ligand activated ion channels. So the neurotransmitter is the ligand, and the ion channel is the receptor. And so when a neurotransmitter binds, this can allow an ion to move out of the cell. That was a bad arrow. Ions can move out of the cell or into the cell, depending on the nature of this ion channel. So when they bind, these ion channels in the postsynaptic membrane can cause them to open, altering the ion flow across the membrane, and ultimately this may result in generating a postsynaptic potential if ions are flowing, right? So this will lead to this ion flow, will lead to a postsynaptic potential, right? Or a change in that postsynaptic potential. Now, this change could be excitatory. So we may have an excitatory change. Let's see. Let me make sure I get this spelled right and in a way in which you can actually read it. So, excitatory. So I want you to ask yourself, what does it mean if the postsynaptic potential is excitatory? That means it must depolarize. Right? Now ask yourself, what could depolarize the postsynaptic cell? Thinking about the types of ions that might be moving. Well, the one that we've seen already is it could be an influx of sodium. Also, calcium are both ions that could be cause the postsynaptic cell to become excitatory. Now we also have the option of it might be inhibitory. All right, and so what does that mean? If the postsynaptic cell, if its potential becomes inhibitory, that means it must be hyperpolarized. All right, and so for a cell to become hyperpolarized, what does that mean? It means that the inside of the cell is becoming more negative compared to the outside. And how would you make it more negative? Well, we might open up potassium channels, because potassium is more concentrated on the inside and it will flow out, causing the inside of the cell to be more negative. Or chloride channels could open and chloride could rush into the cell, bringing its negative charge with it. Both of these would result in a hyperpolarization or an inhibitory potential. And so what are the functional significance of excitatory or inhibitory? Well, an excitatory may lead to, if it's strong enough, an action potential in the postsynaptic cell. So this might lead to a little lightning bolt arrow there, an action potential. Or if it's inhibitory, it'll be more likely it'll be harder to fire an action potential. So it makes firing less likely if the signal is inhibitory. And as we'll see, neurotransmitters, there's a variety of different combinations of neurotransmitters and receptors that can do both of those things. Some neurotransmitters can be both excitatory and inhibitory, depending on the type of receptor they bind to. And then we'll see a whole host of other types of receptors in the coming classes that we would call metabotropic receptors. These don't open ion channels directly and can have a whole host of different cellular effects downstream in the neuron that don't speak directly to influencing how it directly fires an action potential or becomes less likely to fire an action potential. But for right now, we're keeping things simple. Now, to keep these signals brief, I hate how this green keeps popping up. To keep signals brief, precise, neurotransmitters are rapidly removed from the synaptic cleft. Most neurotransmitters, or many neurotransmitters, won't even have the chance to bind to the receptors. And if you, have a little bit of chemistry and cell biology, molecular biology, you know that these ligands, these neurotransmitters, are kind of in a constant flux of attaching to the receptor and detaching from the receptor. So there's lots of trading going on. And so for these signals to be really short and precise, they don't last very long in the synaptic cleft. Right? The neurotransmitters are rapidly removed from the cleft. And this could be done in sort of three major ways. First, we will have what we would call I guess I'll just keep it green here. We might have a reuptake protein in the presynaptic cell. So we'll write these things up here. All right, so neurotransmitter removal from the synaptic cleft. One, we have a reuptake into the presynaptic cell. And that's simply neurotransmitters through specialized channels, proteins getting sucked back up into the presynaptic cell, where they are then available through a cascade of processes to be repackaged into vesicles and retethered in our vesicle synapsin complex. Now, number two, they could be, we might have uptake into surrounding glial cells. So we might have a nervous cell here, a glial cell that's not a neuron, and again, it'll have specialized protein that specializes in the reuptake of these neurotransmitters. And eventually, these neurotransmitters will make their way back, or can make their way back, maybe after some enzymatic processes, back into our presynaptic axon terminal for repackaging. And then third, I'll have to squeeze this in here, we can have some enzymatic degradation And this just means a specialized protein in the synaptic cleft. Let's sort of draw an amorphous kind of blob here. It represents an enzyme that breaks down our neurotransmitters, altering its form so it's no longer the neurotransmitter. It might break it up into multiple different components depending on the neurotransmitter. But this enzymatic degradation makes them inert to the receptor, so they can't bind to the receptor anymore. And these pieces might, in the synaptic cleft, undergo, they might be taken back up into the presynaptic cell after degradation. They might have a sort of a middle cell, so to speak, in the glial cell, in which they can be recycled and made back into the neurotransmitter once again. So a number of options there. But it's important to know that pretty quickly there are various mechanisms to prevent this from happening, or prevent the neurotransmitters from just hanging out in the synaptic cleft, kind of constantly activating these receptors. And then one of our last steps here, we won't forget about our vesicle membrane that fused right here. Oops. There we go, that fused with our membrane. And what happens, we sort of get like a reverse process. Let me match this for thickness, in which the vesicle is retrieved and recycled to make new neurotransmitter-filled vesicles ready to go through the process again. So it keeps that cycle going, right? If it didn't do this, the axon terminal would get larger. The membrane would get larger with each vesicle that merges to it if it wasn't retrieved. And it would become too large eventually. So these vesicles are retrieved from the presynaptic membrane. So chemical synaptic transmission is kind of a carefully timed sequence of events. We have the vesicles that are ready, waiting there for an action potential to come. That action potential causes an influx of calcium via the voltage-gated calcium channels. This causes the vesicles to break apart from the synapsin vesicle complex. It facilitates their movement down to the presynaptic membrane. It facilitates the vesicle's fusion with the membrane, and that results in the contents of the vesicle being released, the neurotransmitters, into the synaptic cleft. They diffuse across the cleft, and they bind to receptors that then become activated. And it causes some kind of postsynaptic response in the cell. In this example here, the postsynaptic receptors are ion channels. So they will open up and cause ions to flow. So they will alter the postsynaptic potential in some way. Calcium or sodium is allowed to come into the cell. Or if there are calcium and sodium channels that open, those ions will naturally come into the cell, flowing down their concentration gradient and electrostatic gradient. This is excitatory. And if that excitatory potential is strong enough to reach threshold, that depolarization gets up to the threshold mark, then another action potential will propagate in this postsynaptic neuron. If the receptor proteins are potassium or chloride, and they open, then that'll cause potassium to leave, going down its concentration gradient, or chloride to come into the cell, moving down its concentration gradient. And this would cause an inhibitory response or a hyperpolarized potential. The postsynaptic cell will become more polarized, making it less likely for an action potential to be fired. Quickly after the release and the receptor binding and postsynaptic potential changing, the neurotransmitter is removed from the synaptic cleft from those three different ways that I went through: reuptake into the presynaptic cell, uptake into a glial cell, or enzymatic degradation. And then lastly, the last sort of step is the vesicles are recycled. They're pulled back out of the membrane and can be repackaged with neurotransmitters and put into the vesicle pool, tethered with synapsin in the cleft, in the axon terminal, ready for the cycle to begin again. So this is a very general. We haven't gotten into any of the molecular details of any of these steps, but this is the general pathway of neurotransmitter release. And we can see the key players here are calcium and a whole host of different types of proteins that we collectively called snare proteins that are involved in the actual merger. Okay, so this ends our quick mini tutorial here on the mechanism of neurotransmitter vesicle exocytosis.